Right, welcome back. Remember, we have been talking about receptors. Now we want to talk about some of the characteristics of receptors, or some of the common terms that you'll actually come across as far as receptors are concerned. Now, the very first term that we shall talk about is what is referred to as spare receptors. Now, this is common, especially among the G protein coupled receptors. And in here, we basically mean that the ability to amplify, duration and intensity of an impulse, for that matter, let's say an impulse, yeah, and yet not all receptors have been occupied, yeah. So in other words, we elicit a maximum response, and yet not all receptors have been occupied. That is what we refer to as the concept of spare receptors. So we also have what we refer to as desensitization. You will find that sometimes, I'll give you a very common example of what we said earlier on. Using coffee, yeah? You drink coffee and then, you do not, the very first time you use it, you stay awake. But then the subsequent times that you use it, you might not necessarily stay awake, yeah? Because of the concept of what we refer to as desensitization. Yeah. So what does that term mean? It basically means that changes, yeah, in the responsiveness of a receptor due to what we call repeated or continuous administration of this particular kind of drug. So in other words, we are saying a time reaches when the cell, or when the cell does not respond as much as it used to, because of repeated administration or continuous administration of this drug. And of course, how does desensitization basically happen? Now, there are quite a number of mechanisms that are put in place, or that explains how desensitization happens. And one of them is what we refer to as, tachyphylaxis. And remember, maybe the other thing that we should mention is that, desensitization is sometimes protective, yeah? In other words, it helps protect the cells or the body itself. Now, when we talk about tachyphylaxis, it basically refers to repeated administration of a drug that results in a diminished effect, yeah? And so this basically means that receptors are still present on the cell, however, they become unresponsive, yeah? They become unresponsive to the ligand. Now, the other theory that is put in place is what we refer to as downward regulation, yeah? So you have, in this particular context, it basically means that, receptors are taken in, yeah? They are taken in. Remember, we said these are macromolecules that are always manufactured or synthesized and destroyed, for that matter. So with downward regulation, receptors are taken in, yeah, by a process of endocytosis. And so a cell where you had maybe about 50 receptors, when downward regulation occurs, then you might have 10 receptors on the cell surface membrane. Why? Because the rest have been taken in or have been endocytosed. Now that is, and so what do you expect? You will not have a response, yeah? You will not have a response that you would have if you had more receptors on the cell surface membrane. And so that is what we refer to as downward regulation. And this also results into what we refer to as tolerance. If you still remember in our very first lectures what we mentioned about tolerance. Now, it is also very important to mention that some receptors, particularly what we call the voltage-gated channels, require what we call a rest, or generally called or referred to as a refractory period, yeah, before they can be reactivated or before they can be re-stimulated. Now, those are the common characteristics or the common terms that you'll find quite often used of receptors. When we come back, we shall talk about what we refer to as the dose-response relationships. Remember, we are still talking about, or we are still handling pharmacodynamics. Pharmacodynamics.